Okay, everybody, very welcome. My name is Hamprecht, and this great fashion gadget here is to help me record this lecture. It's an experiment we do it electronically this time. Some of you were here last year and saw our monstrous setup with the document camera and everything. People who took this lecture course, on pattern recognition, found it helpful to have the videos to look at when preparing for the exams, which we'll have eventually. And some people from outside find it useful also. There are no cameras in the room, so your privacy is safeguarded. I will repeat your questions when you have them, and I like questions so that people out there can follow them also. Okay, very welcome to this image analysis course. Image analysis is very dear to my heart. It's this lecture together with the pattern recognition lecture is those are the favorite lectures I give, and my own scientific path has been quite tortuous. So I did quantum chemistry in the past and data analysis and this and that, and me ending up here means I actually, so here meaning in the subject means I actually like it very much, and I hope I can share some of the reasons why I like it. I am in the physics department, though not located as on the campus, as you have all noticed. The reason was that when we founded this image processing institute a few years ago, there was not enough space on campus. So we rented space out here, and there are now 5 image processing groups on this floor and the floor above. Who among you are physicists? Okay, good. So you must feel really the odd one out here. Okay, so for today, I want to show you 2 or 3 movies of stuff that we are doing in my group. I then want to talk a little bit about the hardware of, in particular, our eye. So we don't really understand how our vision works. But generally speaking, the earlier on in this processing flow, the better we have understood it. And the more interesting things get, the less we understand. So I'll start with telling you a little bit about the hardware of our eyes. I will then tell you a little bit, introduce a first algorithm that allows you to recognize things so that if you decide to never come back again, you will have learned something useful. And then I will give a pretty extensive survey of the rest of the entire semester, also dropping some of the more technical terms for those of you who might have seen them before to give you a better feeling of what's going to happen. And then at the very end, we will find a date that works for all of you for the exercises, which are an important part of this course. So Torben Kröger is your TA, and the exercises will be very closely matched with the lecture. So closely matched, in fact, that Torben will sit in the lecture and after the lecture decide the questions that seemed least understood he will try and put into an exercise. But he's a really friendly guy, okay? So these exercises will be computational in the sense that you will do them with computers. It will be helpful if you are familiar with any programming language whatsoever, but it's not a programming course as such. So we will not do programming for programming's sake. It's more to reinforce the concepts that you've seen here. Okay, so I promised some movies, and here's one that your teaching assistant has produced. This is beautiful raw data originally from Winfried Dank's lab here at the Max Planck Institute for Medical Research. You see a 1,000 by 1,000 by 1,000 cube, so 1 billion pixels of super high resolution isotropic electron microscopic images of the retina. This is quite suitable because we'll talk about the eye today. And down here you see cell bodies, and then up here some of the zillions of processes. These are just the coarse processes that you're seeing here. There are even finer ones, and We've pulled out mainly here the big ones connected with nuclei. On the one hand, to make things easier to look at. On the other hand, because we still fail, and everyone else does too, to get the very fine processes. So this will provide us with work for a few more years. And well, that's a very interesting and challenging image processing problem in itself, to go from the gray raw data to these segments colored by identity. But more than that, the hope is that this will allow biologists to better understand how the retina and later on other parts of the brain are actually working. So this is data set number 1 that I wanted to show. And there is a data set number 2, another big problem that we're working on. This is beautiful data from Lars Hufnagel up here at the Max Planck Institute, sorry, at the EMBL. You see the very early stage of a fruit fly, and because things are so fast, let me stop this for a moment. This is the egg of a fruit fly, so a tiny, tiny thing which, if you let it grow for a day or two, turns into one of these beasts that sit on your apples and bananas if you don't, if you cut them and leave them open. At the very beginning, on the inside, we have egg yolk, and then this manifold replicated DNA on the outside. I cannot really say many cells on the outside because it's only a bit later that the cell walls are actually being formed here when things are becoming a little bit crisper. So now these are really individual cells. And then look, gastrulation. So we have an amazing event. Symmetry has been broken and so on. This has folded onto itself. And now you can stare in amazement how nature does it with what great economy of means and patterns to produce something very complicated at the end. So we see here something has curled around. We see cell divisions going on. You see these tiny bright speckles. Those are dividing cells. Cells are moving about. And if you look from a distance here in the upper right corner, you can already start segments being formed. There's also much going on at the interior. And in the top left corner, you see the time. So we're now 4 hours into the development here. And if we wait a little, we will see the complete larva. And our task here on the image processing side is to track each and every cell. And we will do this in insects, but also in vertebrates. And wow, look at the end here. This is a fast motion here. It's closing up its belly. The organs are developing. And at the end here, it's shaking because it's a live larva. So it means all this imaging and all this, all this light that was pumped into it did not kill the animal. And that doesn't go without saying, yeah? Because when you shine so much light on such a small sample for such an extended period of time, the risk is always to burn everything that you've been looking at. But here you see at the end that actually it's really alive and kicking, literally kicking animal. So I said that we're trying to trace each and every cell in this developing organism because it is not understood to date. If, for example, you take our heart, if all the cells in our heart, if they date back to one particular cell in an early stadium, or if the heart simply recruits whatever is around locally. And to answer this kind of question in developmental biology, we want to track everything. And tracking a large number of objects is hard. Tracking a large number of dividing objects is harder still. And this is why we cannot do it in perfection yet, but we hope to get there eventually. Why is it so hard? Tracking has been worked on for a long time, mainly in the military settings. But in most cases, you have 2 or 3 or maybe 4 enemy aircraft invading the airspace, and here we have 6,000 cells. So that this is why one of the aspects of why this is a little bit hard. The data is amazing. So it's actually 4D data. We have 3 spatial dimensions plus time. Okay. And generally speaking, when working on these problems, we don't try to handcraft them from beginning to end, but rather we try and develop a learning mechanism that will allow you to learn from a training set what's going on. So in this tracking, for example, ideally we want a biologist to say that this cell in the next time step is that cell, in the next time step is that cell, and then it's been dividing into those two, and so on. And we want to use this information to learn a tracking scheme rather than infer rules by hand by looking at this thing and then programming something. Okay. Any questions so far? So this was to give you an idea of what we're working on. Today I will also argue in more detail why this learning cannot be applied naively and why image processing persists as a separate subject besides just pure machine learning.